Okay, so today we have a topic of blood vessels under mammalian histology. Blood vessels, whenever we say blood vessels, in our mind there comes a blood pressure, a very commonly heard term blood pressure. Blood pressure is all the way related with this, very closely related with the artery and veins of blood vessels. It is a pressure of a blood that is liquid on wall of blood vessels. Blood vessels, histology of blood vascular system. Here in this chapter, we'll see how these blood vessels are made up of, what is the tissue structure in blood vessels, what are the different types of blood vessels. Whenever we say blood vessels, in our mind there comes the word blood pressure. This blood pressure is very closely related with the blood vessels. In subsequent few lectures, we'll see the detailed histology of artery, detailed histology of veins, and the connecting tubule between the artery and vein is that is capillary, histology of capillary. Here the terminology is used as artery, vein and capillary, that is based on the origin of that blood vessels. Blood vessels are actually a tubes, a simple tubes connected with the central pump that is heart. From the heart, a tube originates and it goes pass into the body. It goes on branching and branching and branching, and finally where exactly the purpose of blood has take place, that is the capillary, and then they reunite, all the branches reunite and they forms again a single large tube which comes back to the heart, that is called as a vein. So in simple terminology, you can say artery is some a blood vessels which originate from heart and goes towards the body, and veins are blood vessels which collect blood from the body and pour it into the heart. So the blood vessels going towards the hearts, they are vein. The blood vessels originating from the heart, that is they are carrying blood from heart to body, they are artery. And in between very fine branches of these blood vessels, they are called as the capillaries. We'll see in detail that capillary later on. So first we'll focus on our artery. Now just I told about the blood pressure. Blood pressure is we are very much familiar with the blood pressure thing, because in almost everyone we have somewhere heard about the blood pressure problem and the medications and different medical emergencies arise due to the blood pressures. So what is that blood pressure? It is a pressure exerted by internal fluid, that is blood, on the wall of tubes. So you can compare it like in our gardening, when we start a cock of water and we start gardening or watering our garden. That time if we are using some tube, hose pipe, then if you press the nozzle of that hose pipe, it produces high pressure in a wall of that tube, and because of that you can spray the water like a fountain. Same way here, as these tubes goes on narrowing, they goes on increasing the pressure. Here blood pressure in our body is necessary to maintain the flow of blood from heart, and again when the blood comes to the heart, that circulation pressure should be maintained, so the diameter of this artery goes on reducing as it ascends. And Veins, when they come back to the hearts, their diameter goes on increasing, and that is how they maintain this blood pressure in the blood vascular system. Now here in this diagram you can see the arteries, a dark color outer coat, one coat of a artery. If you see start histology, then the outer coat is there, then a pinkish color another coat is there, and another light red color is used for internal one layer. So we can say that this artery, they are made up of three different layers. They terminologically they called as a adventitia, then elastic laminae. This band is of smooth muscles, and internal layer is of endothelium. Now this endothelium word we are going to hear many times, because capillary, they are made up of only of endothelium, nothing this outer adventitia, intima or smooth muscles are there. Basement membrane, internal elastic lamina, everything goes vanished and there remain only the endothelium. Now here they have shown it passing blood from heart, and so it is termed as a artery. Now they produce branches, and the smaller branches of arteries are called as arterioles. Then this arteriole goes on branching, and then they are termed as a capillaries. All these capillary again unite and they forms a venules. These venules unites to form a vein. Again in histology of vein, here you can see they are made up of same three different layers. The wall of these veins are made up of three different layers. But there is a variations in the thickness of this layer. And one more additional thing you see in a vein is a walls. That is, you can say it's a walls present for preventing the backflow of blood. And the arrow is showing that this blood is flowing towards the heart, so it is vein. Now in between this network, between the artery and vein, that is capillary. Now what about the capillary wall? It is a single layer structure, very commonly known as a endothelium. It is such a thin layer that except RBCs, all the nutrients can pass through the wall of this capillary, and that is the site where there is take place all exchange of nutrients and nitrogenous waste material. We'll see that in detail in a capillary histology. Okay, so today we'll see histology of blood vessels, in that artery, histology of artery. Now, though it is artery and vein, but in both the cases, arteries and veins, major artery and major veins, are made up of three layers. Here you can see these three different color for three layers. They are made up of a tunica interna. This is innermost layer. Now these whitish portions indicate the internal hollow lumen of a blood vessels. This slide is a cross section of blood vessels artery. In this cross section, this portion is a lumen, internal portion, and this one is a outermost layer. Now from, so if you start seeing these blood vessels from internal side, the first layer is a tunica intima, that is a very thin single cell layer, that is tunica intima. Thereafter, the middle one is a tunica media. As the name, this media name, you can, it can help you to remember the labeling, the middle layer or tunica media. Now this layer is made up of smooth muscles. The muscular actions of blood vessel is because of this tunica media. All that constrictions and dilation of blood vessels take place because of this tunica media. And the outermost layer is a tunica adventitia. This made up of epithelial tissues that protects or they serve as a covering for the tubes. Now first here we'll see the structure of this tunica intima, the innermost layer. We have another slide where you can see the large picture of this tunica intima. This is innermost layer and that lines the internal wall of these blood vessels. It is made up of a simple squamous epithelium. Now I hope you remember from the first few lectures what is that simple squamous epithelium. Epithelium is a covering kind of a tissue, simple, flat and squamous. It contains layers and a thin layers of areolar connective tissue. Again, you have to recall about the connective tissue from the first few lectures. Areolar connective tissue that form the basement membrane or that stick it to the tunica media, that is serve as an adhesive membrane, basement membrane, which help to stick this tunica intima with the tunica media. Already we told there is three layers. So the internal layer is sticked with the middle layer using this areolar connective tissue. The basement membrane is often seen as a squeezy line. When you see a slide under microscope, this membrane in the basement membrane is not very clearly demarcated. And it shows a right, you can find that basement membrane right exactly right under the innermost layer, that is media, that is tunica intima. When you trace that tunica intima at the outer side, you find a very thin marking of a basement membrane. That simple squamous epithelium can help you to identify this tunica intima. Now, what is the role of this tunica intima? Because of this smooth squamous epithelium, that blood don't get entangled with the wall. It forms a very smooth, slippery coating from the internal side of a tube, so blood can easily slide over the wall. Now, how this tunica intima is originated. So if you remember this cardiac muscle topic from the previous lectures, that heart, the internal endocardium layer of a heart that continues to form the internal layer, that is tunica intima in artery. And now when that endocardium layer enters into the blood vessels, that is artery, now it is called as a endothelium. And you will find that the same endothelium is remain continue when we will see histology of capillary. So in histology of capillary, you will find there is no tunica intima, media, adventitia kind of a layer. There is only a single layer, and that tunica intima is now in capillary called as a endothelium. Now tunica media. This is middle layer in a blood vessels. They are made up of smooth muscles and elastic fibers. And all that blood pressure related activity take place because of these smooth muscles or tunica media portions in a blood vessels, artery or vein. So they are responsible for causing the contractions, that is vasoconstrictions, and dilation, relaxation of blood vessels, that is vasodilation. All these media is in control of different hormones in our body, that vasopressin hormone, their target organ is this smooth muscle layer in a blood vessels. So whenever you get any medicine for blood vessels, that medicine act on either this hormone or that medicine act on wall of the smooth muscles, that is tunica media, and our blood pressure get maintained. Histologically, they are just a smooth muscles entangled with some amount of elastic fibers. Now, green color line portion in a photograph, you can see that is the outer layer of a blood vessels called as a tunica adventitia. It is again the most superficial or outermost layer of a blood vessel. It is made up of a dense irregular connective tissue. You just recall chapter connective tissue, there you can remember what could be the histological structure of this when we say it's a dense irregular connective tissue. They contain lots of collagen fibers, and that collagen fibers, when it's an irregular word is used, that means that collagen fibers are growing to all different directions. In many of the slide preparations, you can see the collagen fibers as swaggy lines in these layers. When we take histology, that is when we take a section of a blood vessels, arteries, that section get cut, that section cuts this collagen fiber, and so all irregularly formed design layer you see as a tunica adventitia. Here in this portion you will not find any pattern, circular, round or vertical or horizontal lines, but they are very irregularly placed structure. That irregularity in a structure is because of this irregular connective tissue get cut during the microtomy, and it look like this. From such appearance of irregular design, you can identify it as a tunica adventitia, and another characteristics, it is outermost layer. Now we'll examine these histological slides of artery under 400x magnification of microscope. Now here that blue arrow, this blue arrow indicating the single thin endothelial layer, or now we say it's a tunica intima. You can compare the thickness of this tunica intima with other two layers. It is almost a single layer. Thereafter, this black color bracket indicate the middle layer of blood vessels, that is tunica media. Okay. Again, here you see the textures of this tunica media. It is a arrangement of a circular muscles, circular smooth muscles arrangement is there. So you find a circular line going on, and this kind of structure when you ever see under microscope, you can say it is a smooth muscles, circular smooth muscles. Then this green bracket indicate the all tunica adventitia outermost layer. Here, because of this irregular dense connective tissue, there is such irregular pattern is appeared. Now you have to train your eyes to remember these structures, how they see under microscope, how they look under the microscope when we see a stained colors slide. In this histology, many times we learn lot of theoretical things, but when we actually see under microscope, we have to correlate that theoretical pattern three-dimensionally here, because it is cut into the single dimensions. So when an irregular bunch of a thread you cut precisely, You may get some vertical, some horizontal, some only pointed structures. Same thing you can see here in this tunica adventitia. It is form an irregular pattern.